Welcome everyone to our wrap-up session for a very wonderful conference. And John Nolan and I are going to be chairing the last bit here. The way the format will be is I'll talk a little bit just about the upcoming meeting about a year from now for the International Carotenoid Society. He's graciously allowed me to talk a little bit about that. Then it will be a question and answer session and free discussion. I'm going to present one case just to discuss of an interesting case with carotenoid supplementation. And then we have another series of questions that have been collected, I think, from various audience members that we'll go through as much time as we want. Yeah. And then we'll just sum up, and I'll give you your homework for the next meetings coming up. So, as I mentioned before, earlier when I gave my talks before, there's always lots more to learn about carotenoids and many great meetings coming up. And so a little less than a year from now, the International Carotenoid Symposium will be held in Park City, Utah. It will be June 29th to July 4th, 2014. This is going to be, it will feature an awful lot of what you've been seeing here in terms of eye and carotenoids, but it will feature much, much more. That will be only about 20 to 30% of the conference. The International Carotenoid Society also features chemists, botanists, epidemiologists, all diseases such as cancer and everything else will also be included. So it's a very, very good meeting and very good interactions that will be there. Park City is beautiful, and it will be held at the Canyons Resort, which is a ski resort. It's very close to Salt Lake City, just a half-hour away. And this is the Grand Summit Resort, the main hotel that it's at. And you can see the ski area behind it. And for those of you who don't know, Park City, Utah is 35 minutes from the Salt Lake City Airport, and it has, as you can see, it's in the western part of the United States. It's very accessible, especially from Europe, with daily nonstops from Paris. And it's very easy to get to from anywhere else in the United States. And there's a lot of things to do at this meeting, a lot of things to do around Park City, biking, golf, a lot of summer sports. It will be up in the mountains, so it will be cool, but very, very pleasant. And there's shopping at Main Street, which is not far away, just an easy bus ride away. And then we certainly encourage people to come early or stay late, bring their families, because there's an awful lot to do at all the national parks. This is a Delicate Arch that's over 45 feet tall, and you can hike to that quite easily. And we will even have, for those of you with families, there will be, it's a ski resort, so there are very spacious rooms to stay in. There's childcare. There's a lot of activities for the family, too. The opening night, we will have, people will go up in a gondola to the top of the mountain to a lodge up at top for the opening reception. The conference facilities are excellent, can hold at least four rooms with 400 people. And we will have posters and everything else, posters and many speeches speakers over those days. And then, as I said, try to do other activities both in and around in the desert elsewhere. So, and then the final night, there is fireworks. There will be on the night before, and also at the grand reception, we will have it at the Utah Olympic Sports Park, where the Winter Olympics ski jumping and luge were held. So that's really it. Oh, and just in terms of the cost, we haven't set it yet. It'll be somewhere in the range of about $650, we think, for registration. Hotel rooms will be about $130 a night. We're getting a great deal for the off-season at a ski resort. So I think that's it. Okay. Are there any questions? There is a website, but it's actually easier to remember to go through the International Carotenoid Society website. Just go there. Fred Kachik runs that. And it has the links to the meeting, and everything will be there. Look for updates. Probably in about 2 or 3 months, there'll be much more detail about the conference. Okay? Yeah. Okay, so it's carotenoidsociety.org, or just do a Google search on International Carotenoid Society. It will come up very easily. Okay, so now is the time for discussion. And I'm going to lead off with a brief case presentation and discussion of this. And the question is, as we've discussed here, we're talking about a lot of supplements and what dose levels to give. And so I'm going to present a case where we might have some ideas as to how much zeaxanthin may be too much for the retina. And as we already mentioned before, there's selective uptake into the eye. We know that just lutein and zeaxanthin make it into the eye. And we know that there's a lot of zeaxanthin, or there's not a lot of zeaxanthin in our diet. Our average uptake is about 0.2 milligrams per day, but it's been studied in a number of toxicology studies, and currently EFSA lists acceptable intake of zeaxanthin at 0.75 milligrams per kilogram per day, which is about 50 milligrams per day. But most trials are using 2 to 10 milligrams. So the question is, can you go too much? And one of the things we want to avoid is toxicity, and this is classic canthaxanthin maculopathy. So it's known that canthaxanthin, a different carotenoid that we don't use very much, can crystallize in the eye in this kind of circular ring-shaped pattern. So we want to avoid that, and the question is, has that ever been seen? And as far as I know, up until what I'll be showing here, that it's never been seen with lutein and zeaxanthin. And this came up in a study that I've been doing, a small clinical study for MacTel, Macular Telangiectasia, which is a rare disease where there are abnormal vessels that accumulate in the retina and pigmentary changes. And one of the things that's been found in macular telangiectasia type 2 is that carotenoids have a maldistribution. That's been well known. And it's shown actually quite well here. I think you should be able to see this in this autopsy eye, that instead of a central spot of carotenoids, as you would see, the central macular peak, you actually get a big ring of this. And you can see in this patient here, in the autopsy eye here, this ring. It goes almost to 7 degrees here bilaterally. And no one knows why this occurs. We think it may be a maldistribution of the binding proteins, but we really don't know. And this raises the question, as we're all trying to change the central, can we fill it in by giving either lutein or zeaxanthin to a patient with MacTel to change this distribution? And previous studies have tried this with lutein, and it didn't change anything. But no one had ever tried it with zeaxanthin. So I proposed to do a study looking at zeaxanthin in 10 MacTel subjects, and we actually are up to 10 now. And we were going to give them a high dose of zeaxanthin, 10 to 20 milligrams per day, and evaluate every 6 months. And this was IRB approved. It's FDA. I had an IND and everything else that's required for this. And shown here is one of the patients who was in the study. And this is her at baseline in January 2012. And this is the picture here, shown here. That's the initial picture here. And you can see that in this, the macular pigment in an autofluorescence image is a dark circle, as you would expect, consistent with this disease, not the central spot. And she came back after, I think, 6 months on the study, 20 milligrams per day. She felt she was seeing better. But there was new things here. You can see right here. There are crystals that have occurred in her retina right there. Not where we would expect them to be, not at the edge of the ring here, but there are crystals. And you can see that we did raise the amount of zeaxanthin in the retina. You can see that the circle here in her right eye is much, much darker. The circle here is somewhat darker. And you can even see the crystals here, as would be consistent with an autofluorescence picture. The crystals are dark. And these were golden yellow and somewhat refractile. So this, to me, was the first sign of a possible crystallization of zeaxanthin in the retina. And we got an OCT shown here right through the crystals. And the crystals are right here in the inner retina, not in the fovea, but right there. You can see it in that region. So the question is, okay, and this is open for discussion, what would you do next? Okay, you're running a clinical trial, and we've seen this. So I will open it to anyone. The choices are continue supplementation at the current dose, decrease the dose, discontinue and see if they disappear and maybe restart it, stop it all, or stop the whole study. So, yes?
Did she increase in the center?
No, she did not increase in the center.
Yeah, Stephen?
Paul, how sure are you that this is anything to do with the supplementation?
The main thing that I can say is that I had followed her for many years. She'd been a patient of mine for probably 8 years, I think. She did not have these crystals in February. As soon as I put her on the supplement and brought her back, they were there.
But if we go back to the OCT in the photo there,
Yep.
again, the photo, as you say,
That's
it's almost outside the macula.
Yep.
So it's not only outside the area of macular pigment, it's most certainly even further outside the area of zeaxanthin distribution. If we go to the OCT.
Yep. And let me just say that there were, when you looked hard, there were actually crystals in the other eye too, around the that were at the edge. You maybe see one or two there.
Could we go back to the baseline pictures
Yep.
then? Was there anything there? No. Okay. And then if we go to the OCT.
Okay.
Do you not think it's too superficial there for a distribution of macular pigment? I mean, that's...
I agree it's
there.
in an unusual spot, but that's where it is. And that was, we have the OCT from before, we have OCTs, and I have the OCT, you know, after this.
I mean...
It's really this ring-like distribution, so they may be able to...
Can you Tuss, do you want to take we can't hear you. It's right there.
They have this ring-like distribution, so they may be, instead of us, store their macular pigments in the ring instead of in the very center.
Right.
Because in some subjects, I've measured a lot of them myself too. It's even higher in this ring than in normal control. So it's not that it looks like that it's really different. It's not normals having a hole in the middle, but something different.
Yeah.
John?
So Paul, you know, I'm thinking back to gold dust maculopathy, which was really a hot subject for a while in the 80s, and the outcome of that was discontinuation caused loss of these crystals in the retina over time, and in fact there was no pathology associated with this occurrence. It was odd, it was perhaps worrisome, but in the outcome, the final outcome, the subject's vision was not negatively influenced. And so with this history, I think the difficulty is working with an IRB board, which is of course very conservative. And I know mine would say E would be your only option. You got to stop this completely for all subjects. But you know, I think with what we know and the perspective of this community, one would say, you know, you just want to pull back and see if you get a recession of the crystals and improvement. And then I'm going to add one thing, because of course I assume you're supplementing with RR zeaxanthin. You know, we're a little loose with our terminology, as Neil pointed out.
Yep.
And I'm wondering, you know, RS zeaxanthin is a diastereomer. It's not an optical isomer, it's a diastereomer. So it is a molecule with quite different physical properties in many ways. So some of the times we say, oh, they're identical, but no, they're diastereomers. They have different dipole moments and other factors. So a mix of RS zeaxanthin might well cause non-crystallization to occur. A mixture might not crystallize, so that would be an interesting follow-up.
And actually, if you compare, and I don't know this, if anyone knows this, is RR, pure RR, does that crystallize easier than RS, or are they about the same?
No, don't go there. It has nothing to do with that.
Just
Okay.
for the benefit of the non-chemists in the room, RS is meso and RR is the other zeaxanthin.
Yep. Okay, go ahead.
Can I just ask?
Yeah. We have one there, and then Fred.
Have you done 2 wavelength autofluorescence just to check it's got the right spectral absorption, that it really is?
We did, yes, she does. She did have, I'm trying to think if we had that. We sent her over. I'm not sure her fixation was good enough to get a high quality. Okay. But I'm pretty convinced that this is.
That's what it is.
That, you know, this was new. It had the right color. I mean, I looked at it directly. So, yeah.
Paul, did you do the blood analysis on that subject?
Yes, we have that.
Anything abnormal about the levels of lutein, zeaxanthin, and other carotenoids, or?
Not that I know of, but I can review that. I just don't know the answer.
And also the dietary intake and all this other stuff. You know, I've always wondered about interaction between these things, because when you give megadose of one compound, you got to be careful with the other interaction with the others.
Yeah.
And the other point is that as soon as you start interrupting the normal dietary ratios of these things, you're really messing around with the normal metabolic pathways.
Yep.
That's the only comment I have to make.
Yep.
Just
Normal values, they have no compromise in gut or so. Serum values are similar between controls and these MacTel subjects. So
Yes.
in
yeah.
general.
Yeah. There's nothing unusual.
Were there any visual disturbances as a result of it?
She felt she was seeing better.
She felt she was seeing better.
Yes.
And
I could not document that, but she
and
felt she was.
Presumably, if it's crystallized, dual wavelength autofluorescence won't help because surely the spectral absorbance would probably have changed.
Well, it'll give you two different spectra. That's it. Yeah.
Yeah. So that's and one last question. MacTel, as you say, is a rare disorder. Is there in the natural history of MacTel, do any such lesions have they ever been reported?
So in MacTel, there are crystals that are present, but the crystals are probably do not have the right spectrum on autofluorescence to be and they're smaller and they're closer into the fovea.
Well, in answer to your question, what I would do is I would stop this patient for a few weeks and reevaluate then. That's what I would do.
So that's a vote for C, I think. Yeah.
C.
Okay.
Yes, I guess that your patients had some intraretinal cysts before the supplementation.
So, I'm sorry.
The cyst, intraretinal cysts. Cysts.
Yeah.
And
actually, very few, but
very few.
Yeah,
and
you can see her OCT. Her other eye was worse, I think.
Yes, and you didn't see any change with this comparing after and before supplementation. Okay, thank you.
Okay. Any other comments here? So, what we did actually is anyone else want? You can make a comment. Oh, I think there's more. We have time. We'll do just one or two more and then we'll move on.
I think so. Good.
Yeah.
Did you do some ERGs, multifocal ERGs?
Yes.
And they were normal?
They were normal, and we did, the only other thing with microperimetry was very abnormal in that area. So we'll move on. Yep.
And we have one last question. Lee.
Did you classify the adverse event as serious or no?
We classified it as just an adverse event. I reported it to the IRB and to the FDA, and we went with option C. She was the only one discontinued because we re-examined all of our other patients, and they had no signs of crystals. And she came back 3 months later, and the crystals were gone. It took only 3 months, which is very unusual, at least compared to canthaxanthin. And she asked to go back on zeaxanthin at this point. She felt her vision got worse and went back at the 10 milligram dose and came back. I guess I last saw her in February. I think she's due to come back soon. And the macular pigment is coming up again in the ring-shaped pattern, but no crystals this time. So that's it. Okay. So next here.
So the next thing these are the questions that were submitted.
Yeah, if you want to do, you can lead this part.
Okay,
maybe.
right. Cool. All right, so you can see them. Let's have a look at them. What does AREDS2 mean for macular carotenoid research? Does anyone want to take that on? Any comments on the implications of the AREDS2 for what we do?
What AREDS2 does for macular carotenoid research, staying to that specific question, not practice, macular carotenoid research, in my view, that the standard of care is now AREDS2 formula. So that's the AREDS2 formula minus beta-carotene with lutein and zeaxanthin. So I don't think in a clinical trial that using a placebo other than that is justifiable. That's my view.
Any other comments on that? Yeah, Lisa.
I think one thing that it means for our research is something that we've seen at UGA over time, which is that it's getting harder and harder and harder and harder now that more people are taking supplements to study true 0 points for lutein. So I think that if this does in fact become standard of care, as Stephen was saying, then what it means for us is that as more people take it, finding folks who are naive to supplementation for research studies is going to be a tricky thing. Yeah, it's better for the populace, but certainly I think the way that we do clinical trials may have to change if more folks are actually on it.
Yeah, I mean, and consistent with that, one of our early findings was the offsprings, the sons and daughters of people with macular degeneration had a very clear, significantly less macular pigment level. That's changed now, And the reason why that's changed is you take these people in, measure their pigment, they're all supplementing. So it's absolutely shifted. So I agree with you. Any other comments on that first question?
I do have one. As an AREDS investigator and AREDS2, and as Emily Chu mentioned, there's still some controversy among the investigators. So there are a few very vocal investigators who you may see, and coming, who maybe will invite that person here someday, who very strongly feels that unless you're a smoker or maybe a former smoker, that the AREDS1 is the proper supplement, and is very adamant about it, and is going to make actually probably a lot of trouble for the upcoming papers. I think she'll be writing letters to the editor. Yeah, it is politics. I think, and we can have comments from the people who are here from the supplement industry. I think that an AREDS1 supplement will become very rare. I don't think there's going to be much demand out there, so it may become sort of a moot point.
Okay.
Yes, Fred.
Fred, yeah. We're just going to get your mic.
Here's what we know. In fruits and vegetables, the levels of zeaxanthin is not as much as lutein. But when we look at the blood, the levels of zeaxanthins is elevated, higher than what is in normal diet. Therefore, what I would suggest, if there's any ratio to be supplemented, is the ratio that we could use from the blood. In other words, if somebody was on a high diet of lutein and zeaxanthin, you wouldn't tell them to take supplements, right? So if there is any ratios to be suggested for formulation, I would suggest the blood ratios, and we have plenty of patients or people that have, or healthy people that have a ratio, certain ratio of lutein and zeaxanthin. Coming up with a sort of an average of the two is not that hard. So I really am against the fact that you want to sort of manipulate that ratio to a point where we don't have any scientific evidence for it, which is the normal metabolic pathways. And then include also mesozeaxanthin, which I'd have no idea how the body is going to handle that. But
just a comment on that. I mean, in terms of trying to do what's natural and follow ratios, I think as soon as you supplement, you're doing something that's, you're changing what's normal. So regardless of the supplement. So I think that would be my comment on that. Any other comments? Okay, we'll move on. Again, measurement of macular pigment. Which technique? Let's split this up into which technique for scientific research, clinical trials, measuring macular pigment for correlating. Any comments on that, based on what we've learned and what we've seen?
I personally like imaging-based methods. I think it's very important to really look at the distributions. And I know, this is my bias. I think that the psychophysical methods can do it, but they're only going to be a couple points, and you can get so much more data from imaging-based methods like autofluorescence. And I know that there are issues with autofluorescence and cataract, but I learn as an ophthalmologist, I learn a lot looking at these images all the time.
I think Paul, you're absolutely right on that score. But there are a lot of research groups that are not clinical in the sense that they cannot dilate. They don't have access to the expensive imaging instruments that might be necessary to reach this standard. And so a lot of the nutritional research, which is going to be done with still relatively small studies, and in groups that don't have this kind of expertise, are going to need an approach that is competitive in some way. And so I think it's very important that we figure out ways to lock the measurement of one type of technique to another, so that we know what we're measuring in each case and have crossovers.
Yeah, I mean, and I think that's why I wanted to split that answer up into what we do in the laboratory versus what can we recommend to primary eye care professionals, optometrists. Because, the UK, they've been asking time and time again, what should we use, what should we recommend? And they've had some really bad devices, and it's really done damage, I think, to actually measuring macular pigment in that important way. And so I think it's ongoing. I know there's various flicker devices there. I'm aware that Heidelberg are also looking at their device to make it more feasible for the optometry clinic. So I think it's a debate that will be ongoing maybe.
Yeah. There are ways, and it's Werner Gellerman, who I'm working with, and does have some, there are ways to do autofluorescence without dilation and at lower cost that he's trying to develop, and that may be available in a few years. It's not available, not widely available yet.
My experience with that, and when we got the new Spectralis in Waterford, we were told that you don't need to dilate. And we tried doing it not dilating, and it wasn't working for us. It just wasn't working. And so we made a decision that we absolutely have to dilate.
And I agree. With the Spectralis you can do an OCT easily undilated, but for autofluorescence, you do need dilation.
Yeah.
And so.
Any further points? No?
Well, this reflective techniques, this absolute reflective technique, is being commercialized. So in 1 or 2 years, maybe there will be a device in the market which gives you the ability in a second or so to have a number, but until then, I'll go for the autofluorescence as Paul suggests.
And you suggested in your lecture that you can do this very, very quickly.
Indeed. Yeah.
Yeah, you can do it quickly.
A few seconds.
But it takes longer than a few seconds to get things right, to get the patient right.
Well, let's say a minute.
Okay.
The actual measurements is a second, but you have to sit down and look into a bright light, and then it's done.
Okay.
But is your method imaging based? I know you can do reflectance, but
that's not image based.
That's not image based.
No.
So that's going to be spectral based.
Then why would I go for the autofluorescent? Definitely.
Okay.
Yeah.
Dr. Howard?
Yes, I think that looking at the various machines, of course, some are better than others, but I think you need something that measures the density at 0.5 and 0.25, because you get this sort of central dip. And I think we shall find in the future that this central dip is much more important in its relation to AMD or other abnormalities. And therefore, I was speaking to Richard Bone, for instance. I said, well, could you modify this so you could do it at the 2 points? And he said, yes, we could. Now, I think that maybe that's what we should be aiming for. You can do it with the densitometer, and you can do it with the Spectralis, but I think other machines should try and emulate that.
Okay, very good. We'll move on then. How much, I think we've touched on this already, how much LZ and MZ do we actually need? Do you feel we've covered that already, Paul, or is there anything else we can add to it?
It's been covered heavily. If people want to make any other comments, I think obviously we don't know yet. That's kind of the bottom line is the answer, and we do need head-to-head trials. We do need we can do these on relatively small scale. You don't need to do an AREDS2 type study, and you can, as long as you're doing it in young people, you can still do placebo-controlled trials and everything. And that I agree that in AMD patients, it's probably not ethical, and you probably won't get it through an IRB anymore with a true placebo. But it's important from a scientific standpoint to at least do some pure trials, and at least small-scale pure trials to really to answer some of these questions, and then we can extrapolate it. Yes?
To a certain degree, I disagree to this opinion, and the reason is the following. When I was young, it's a long time ago, you might argue, but nevertheless, the average ophthalmologist was not aware of evidence-based medicine. Nowadays, when people treat patients, and this is a development of basically the last 20 or 25 years, everybody tries to stick to evidence-based medicine. This has completely changed. But what we must not forget is that evidence-based medicine is very limited, because there are very many questions that we cannot really answer in clinical trials. And the AREDS2 study is already a sign of this, because even this question, which does not seem to be so extremely complex, and they had a very high number of subjects and a very large amount of money that was spent, did not finally answer the question. And this is also what you referred to, why there is disagreement between the different investigators, because at the end of the day, the primary endpoint was not significant, although I agree that the study shows it. But nevertheless, this shows the problems of evidence-based medicine. And if you now want to go really into clinical outcome studies that compare lutein and zeaxanthin, maybe also at different dosages in terms of clinical outcome, this is simply not feasible. And
Right.
I think this is a thing that we must be very clear also in terms of the budget situation that we have nowadays, that there will be no large clinical outcome trials that will finally answer this question.
No, I would agree with that. What I'm kind of talking about is targeted trials to answer some smaller questions. Right now, we're with the AREDS trial, where the standard of care in the US and Europe and everywhere else is going to be the 5-to-1 lutein to zeaxanthin ratio. Is that really the best? Well, what if you did do a 1-to-1? At least if using macular pigment distributions and can be a surrogate outcome, and you can do small trials and at least.
You can do small trials on surrogate outcomes. I agree, but we will not be able.
No, there will be no AREDS2. I don't.
So we're going to go Randy and then Julie.
So I think I would rephrase this question in terms of, what you need from supplements and what you need from food. So if you think about the diet that most humans had for 50,000 years, there could easily be 100 milligrams of lutein and zeaxanthin in your diet in a given day. If you look at the paleo diet, which is mostly plant-based whole foods, there's some cultures that have 20 milligrams a day just as an average intake. So if you have a good whole food plant-based diet, you get what we would consider megadoses of isolated lutein and zeaxanthin. And I would argue that that's what you need. You need to match the diet that our bodies evolved to have, and then all this talk of supplements is kind of moot.
I agree.
Julie?
Yeah, I think they're, given the the cost of clinical trials and the lack of opportunity to do big ones again like AREDS2, and also some of the ethical concerns now that there's a convergence of the body of evidence for potential protection, we do have a high rate of supplementation, and this is an opportunity for observation. We can observe people over time, so creating networks and databases so we can use what's naturally occurring in the population and collecting the data so we can evaluate that in lieu of a trial, and there are some statistical techniques that can be used to do adequate comparison when there isn't an experimental model. So I think that might be an opportunity to learn more.
Okay. Any final questions? Okay. Z equals MZ, question mark. So Stephen.
Well, I mean, Liz Johnson and Martin Uringer have eloquently shown that when you supplement primates with lutein, it's converted to mesozeaxanthin. And you supplement carotenoid deficient primates with zeaxanthin, it's just zeaxanthin you find. So I think by definition, from the results of that study, zeaxanthin and mesozeaxanthin are two separate things. But ultimately, it's a question for a chemist.
Yeah.
So maybe Fred, do you want to answer this?
Hello. I'm next.
Oh, sorry. Yes, Dr. Howard. Yeah.
I think the answer is that this should be one of the points for the homework, which Professor Bernstein is going to tell us, because the answer is we don't know, and it's one of the things which we plan to do in our work is now to compare MZ with, or MZ as you may call it, with the RRZ. And I think that we think there's a difference because it's mainly the MZ which is in the center of the macula, and that Z tends to be more towards the outside. Whether this makes a difference, we don't know, but we think it might. And therefore, a correct comparison is the only way of actually doing this, and it should be done.
Thank you. Neil? Have we mic? Oh, Fred has the mic. Yeah.
One of the things that I have not heard at all about in all of these clinical studies is how people look at the metabolism. I mean, what happens if you give lots of mesozeaxanthin? What happens to the oxidation products of mesozeaxanthin as opposed to giving zeaxanthin or lutein? We need to understand, because these oxidation products actually end up eventually in the retinal. We know that. And is there a difference between oxidation products of lutein versus zeaxanthin versus mesozeaxanthin, because they're going to end up eventually in RPE choroid? Perhaps some of them will be transported into the macula. So without understanding the metabolism, you really can't just go ahead and load up somebody with mesozeaxanthin, zeaxanthin, and lutein. The other comment that I want to make that has not been really addressed is that nobody so far has taken pure mesozeaxanthin. And in fact, the reason for that is probably commercial points, because commercially lutein is converted to mesozeaxanthin in certain ratios. That ratio is then taken up and mixed with other things to come up with a novel ratio for all three carotenoids. And I don't think this is the way the science should be progressing. I think you need to do individual studies, understand metabolism, distribution, not only in blood, but of course, hopefully, there are other techniques that can be looked at. But just come up with a ratio based on what you make in the lab, I don't think is a right approach.
Yeah, I'd like to approach it a little bit differently. There's another carotenoid, astaxanthin, which also has 3 isomers. And if you refer to something as astaxanthin, you're not referring to either the RR, the RS, or the SS. In the analogy you have up there, you have one, as John corrected me, diastereomer, the RS, but what is Z then? That doesn't specify what diastereomer you're referring to. There's 2 other ones. And another example is alpha tocopherol. There are actually 8 isomers of alpha tocopherol, but whenever you refer to alpha tocopherol, it refers in general to all of those, unless somebody says RRR, and there's a carrier protein specifically for that. So I think if you're going to use the term zeaxanthin, you have to be referring to all 3 of those diastereomers. If you use the terminology that distinguishes one diastereomer, MZ, you have to be using not just Z, you have to be using the RR or the SS or the combination, because that Z right there doesn't specify what other diastereomers you're referring to. So in general, and I know this group is oriented toward the macula, but in general, if you're referring to a carotenoid by its general name, zeaxanthin, that's going to refer to all 3 of those diastereomers. If you refer to astaxanthin, that's all 3 of those. If you refer to alpha tocopherol, that's all 8 of those. It's only when you refer to, in all your publications or on your label, if your label says that it is RR or that it's RS, then you can say, oh, well, this doesn't contain X amount of zeaxanthin, but if they haven't specified on the label that it is meso or that it's RR, they just say zeaxanthin, all 3 of those optical isomers diastereomers are part of that.
John?
Yeah, I think, Neil, that's a really great point, that clarity in communication and labeling is really lacking. And it would be helpful to us all to be specific. I think that this, natural laziness to use Z comes about because higher plants produce exclusively the RR isomer. And so the zeaxanthin that might be derived from, say, the marigold or some other higher plant source would naturally be the RR. But I think you're absolutely right. We've got to start labeling, because now commercially MZ is available in abundance, and that can be left off in commercial transactions, and what starts out as labeled MZ RS could be sold and become suddenly Z. And so I think it would be very helpful to us to be more clear in the labeling process and the communication. I want to address this question of the two molecules, because as I mentioned a moment ago, they really are distinct molecules. Their dipole moments are not vastly different, but they are different. These are not optical isomers And so we would expect their melting points to be slightly different. We would expect their solubilities to be slightly different. We expect a lot of things. Optically, the UV visible spectra will be identical. They are the same chromophore in that regard. But we do expect them to have properties which could be utilized in terms of binding to proteins. There is some difference, Paul has seen, in terms of binding of zeaxanthin to the protein.
Yeah, but it's surprisingly small.
It is surprisingly
unexpectedly small, but that's consistent.
But what I was going to say now is that what we don't know, your images show us very wonderfully where the protein is. But what we cannot do, and what we'd all love to do, is have an antibody to the carotenoids themselves and be able to label the retina and image where the carotenoids are. And we don't know what percentage of the carotenoid is bound to the proteins. We don't know what percentage of Z is in what areas, in what cells, and in what organelles. And so I think those are questions that remain to be addressed, because if MZ is produced in a particular place in the retina, it may have a particular unique function there. And so those are questions that they're going to be very elusive. They're going to be hard to answer. But I think they're things that we want to address because they are unique molecules, and these three are all there. Do lutein, zeaxanthin, or R-zeaxanthin, and mesozeaxanthin have distinct functions because they are in distinct places, and not necessarily because they have distinct chemistries, but because they're localized differently.
Yeah, I'm sorry, in response to that, I'm not going to argue whether there might be a unique function or something like that, because it's very possible there could be. We know that there have been drugs that are produced, like thalidomide, where one form is healthful, one form is going to kill you. So it's not a matter of functional definition in this case, because I believe that it's very possible there is a different function, the same as there's a carrier protein for all-RAC alpha tocopherol. There could be something here. But if you see somebody refer to something as zeaxanthin, I mean, that includes all three of those diastereomers, the same as if somebody just says astaxanthin to me or alpha tocopherol. It doesn't specify. And I think on a label, if it just says zeaxanthin, by that nature, you have to assume that's what's there. If the labeling or if the paper goes to the point where it's identifying all the different forms, then I think it's appropriate. You discriminate. They are different.
Okay, Lisa?
And I think the other thing to say here is that, the debate over whether or not there are different properties when these molecules are in retinal tissue is definitely one that should be solved for all sorts of reasons. But the bigger question of whether or not to feed all three of these molecules is a totally different type of question. As Randy said earlier, and I have to agree with this point, one thing that I heard yesterday when I was listening to the AREDS2 discussion was a constant sort of accidental statement of the word drug instead of supplement. I think one thing that we do, that we're sort of, a road that we're sort of heading down here as a group, is looking at these things nutraceutically. We're applying these molecules, specifically this molecule, to a disease and forgetting that there's an entire lifespan of behavior that went into acquiring the disease in the first place. I can say that in the United States, I work with a lot of healthcare providers. I work with a lot of, patients, and what I see are people who don't eat well, they don't exercise, they have an entire sort of lifestyle that has led up to that point. So now we're discussing, and I think as a group this is something to consider. We're now discussing whether or not we should be feeding people something that, with the exception potentially of fish skin, we haven't been, and I'll leave that debate again to the folks who are actually analyzing those tissues, but this is not something that is all that commonly consumed, if it's consumed at all. So it's not something that has historically necessarily been in our food supply, but we're now advocating potentially supplementing it. So I think maybe another source of debate that should be considered by the group at large is that issue, independently of whether or not there are functional differences as a result of having these multiple molecules via whatever conversion process they come to exist in retinal tissue.
Yeah, I think, if the view is, and we've kind of touched on this already, if you want to do something that's really healthy and run marathons and eat lots of spinach and all that's where you want to be at. But the reality is different. So I'm just trying to push that point that, you know, if you want to try and do something that's, if you believe that augmentation of these nutrients in the macula is important, well, okay, there's different ways you can do it. Diet is one, supplements are other. I think you have to look.
You say diet is one very dismissively. So yeah, diet is one.
No, I don't say it dismissively at all. I eat healthily. It's what I do, and it's the recommendation. It's the recommendation. Steve, first thing he says to the patients, healthy diets, smoking cessation, and this kind of falls into the next point. Recommendations for the optometry. What should the recommendation be? So Stephen,
can I just make one final comment? I agree with Neil. When you say zeaxanthin, it refers to all 3 stereoisomers. And I think we should be honest with the public. When we're producing a dietary supplement, you need to say on the label what it is. Yeah, and you also need to say mesozeaxanthin is not naturally occurring in the diet, but is a form that may be beneficial to your eye health. And if the consumer has that information, they're not chemists. They don't have any clue what the structure of meso looks like versus zeaxanthin versus lutein. So you really need to, if there's any labeling laws, I guess there isn't any for nutritional supplement. You can say anything you want. But I think the public needs to be informed what it is that they're taking. As an optometrist, for your next subject, the optometrists that are not chemists need to be informed what mesozeaxanthin is.
Equally, and on the same philosophy, I don't think you can say that mesozeaxanthin is not in the food chain. It is in trout flesh. It is in salmon skin. Salmon skin, it's only in the Western world where it's not eaten as a matter of course. And in fact, we found it in sardine skin, and you can't eat a sardine without eating sardine skin. So to say it's not in the food chain is just factually inaccurate.
You could say there is 0.1% of mesozeaxanthin in your diet, but do you want to take 5 milligrams or 10 milligrams a day?
But that's the point.
See what the public decides.
But you were advocating specifying that it's not in the food chain. That would be inaccurate.
But I think the point is that in the United States and probably here, that 99% of people probably don't consume mesozeaxanthin daily.
Probably, but you still couldn't state that it's not in the food chain.
Okay. Any other comments on the optometry point, the recommendations?
Just on the subject of diet, and going back there, one of the things I think that can be easily overlooked is the whole subject of biofortification and bumping up the levels, like we've done with the sweet corn. So it's one way to get around not eating enough is to pack a lot more punch into one go, and we're yet to test all that against supplements, but I think I'd just like to bring that up as something rather than, because nutritionists often say, oh, you mean there's more than one sort of corn? We've never heard of that. There's different types of tomatoes, that sort of thing. I just spoke to a nutritionist and she didn't realise that you could get variation in these things, but you can. As a plant physiologist, there's a huge variation. The genetics is there to be tapped. So.
That's point. Graham?
I'd be interested to know actually how many optometrists are here today. I know there's at least 2 of us.
Have we optometrists here? Yeah, lots of optometrists.
So we are outnumbered, but not totally. That's interesting. I'm not sure if I was dozing off there, but do we actually get any recommendations from you people for what we should be doing?
Is there a consensus, Paul?
I don't know if there's a consensus. We have. I think that, as this meeting shows, that for optometry, that we, just like with ophthalmology, we need to increase the awareness. AREDS2 gives at least a little bit of information, but optometry is very much a primary care type, and you're dealing with the patients, not only with AMD patients, but with their children and the worried well. And you still have to do just what I do in my practice, which is counsel people on diet, give them a handout to try to educate people to definitely follow what evidence-based information we have in terms of making sure that patients that are in the intermediate to advanced stages of AMD are on their supplements, and then talking with the patients. I mean, I've prepared a handout, and I think that there are many optometrists that have done the same, and to emphasize the importance of nutrition, whether dietary changes or taking supplements if you can't change the diet and you need to do it.
Yeah. Of the optometrists here, how many of you actively manage risk factors? Do you look at all the risk factors and make recommendations? Graham again?
That was the answer I was hoping for, but the answer I was expecting to get was for people to say, go and buy a spectralis, go and buy a MP machine, sit your patient down for 10 minutes, 20 minutes, 30 minutes, and send them out the room and let them be happy with an MP measurement. I'm glad nobody actually said that. But the truth is, in the UK, and I'll probably be accused of being a whinging optometrist, but in England, the NHS pay for 10 minutes per patient per year. Once you spend more than 10 minutes with your patient, you're generating negative profits. It's different in Scotland, it's different in Wales, but time is everything in the UK. What I think you should be telling us optometrists is don't try and fit it within your normal optometry session. Lift the whole thing out. Use MP measurement as part of the treatment, part of the advice. Do the whole thing and do it properly. And as you rightly said, go through all the risk factors, go through all the diet, but don't try and add it on to what you're already doing. Lift it out and do it entirely separately.
Okay, I think have we any? Oh yeah, wait, David. Sorry.
Just one point about.
Could you hold the mic to? Yeah.
Sorry, just one point about the handouts that medical profession give to patients. They're usually very poor in terms of information. I mean, on the first day of this meeting, you showed the amount of material that was present in spinach and various other green vegetables. People in the community who are not nutritionists need some advice on how much spinach, how much broccoli, how much of these various quantities, to get some real idea of what you mean when you say increase the vegetable intake in your diet.
Exactly, what does it mean? Yeah. Okay, I think Robert has one, and we leave the final point on this with Robert.
I might have a question here. It's connected with the question how much LZ and MZ. It looks like difficult for the group to decide how much really has to be recommended or how the diet has to be changed. But we are measuring now the macular pigment. Is the group able to give an advice? Today I measured one person, doctor, on my device, and he has no macular pigment. Do I have to tell him that it's good, it's bad, or to what macular pigment you should change your diet or your supplementation? Just a simple advice. What is a goal for a macular pigment value for this person?
So the point is, Robert measured someone today, they basically no macular pigment. What should be said to that person? Right?
It's not the first person.
Lots of people at this meeting. That's the surprise of low macular pigment levels.
Did you question these? When you see someone with no macular pigment, how often does it correlate with a poor diet, or is it a total surprise to them?
It's a total surprise.
Okay.
He's an ophthalmologist, so he is in the topic. I know other professors from the field who are really doing research in macular pigment, so don't definitely.
Yeah, I mean, I would certainly, as we know with MacTel, there are, I would certainly encourage, in addition to counseling them on diet, I would certainly encourage them to have an eye exam. They may have something going on. I think, even I can remember back in the early days when I was doing Raman spectroscopy measurements, there were one or two that came through that had very low pigment, and they were the MacTel patients. We didn't know, I never published it, I didn't know the significance of this, but those patients can show up in your practice. It's more common than we think. And so, I mean, I think that it should at least be brought to the people's attention that there's something going on. Unless they tell you, I never eat fruits and vegetables and I smoke 2 packs a day, then you know the problem.
But maybe even having a good, proper diet, as Julie showed, if you have wrong genes, then you may end up with a low macular pigment because of other factors. So it's not diet only.
You see it the other way as well. I've seen the overweight person who smokes a lot of cigarettes that lives on chips and their pigment is 0.6. I've seen, if you measure a lot of people, you see all sorts. But we make recommendations based on averages.
It's probably corn chips.
Corn chips. last comment with John then before we move to the final.
So who would have known that that little separation that we did back in 1995 and discovered this second pigment in zeaxanthin would have caused all this trouble. But I still, and not being a nutritionist, not being a clinician, see this in a very simplistic way. And that is, there are 3 carotenoids in the eye. And if we have an individual who doesn't have all 3 of them, and one is going to advise them in the most expeditious way to potentially alter and come back to a, quote, more normal macular pigment level, one would be advising them to take a supplement that has all 3 of these. But the fact of the matter is, if they're low and they're not eating these carotenoids, we should be advising them to eat carotenoids. AREDS says they're effective for AMD. It certainly is true that they screen for blue light. So that should be generally protective for the outer layers. So whether it's a lutein only, an RR zeaxanthin only, an RR zeaxanthin and lutein only, or a lutein and meso only, you're going to do good with a supplement. I think you're going to replete the macula to some degree. You're going to improve blue light screening at the very least. Whether you solve the little dip or whether you don't, I think those are questions we don't know the answer yet to, and we'd all love to know those answers, but until we do, we're not going to be able to specify the exact perfect choice. The fact is, though, that these carotenoids are clearly good for healthy vision.
Good. We'll leave it on that. So for the last part of this session, we're going to go back 2 years and see if Paul's carotenoids have been working. Let's see how he's aged. We're going to see the last bit of homework we got, and then we'll create some more.
Being the keynote speaker, I get 2 minutes to sum up, right? Okay. So what have I learned here, and what do we need to do next? And we have a charge, I think, from the Howard Foundation people and the sponsors here to come back in 2 years and have something even more to show for us. So I've just kind of thought of things that I need to do, because I've thought of a lot of new ideas, and I'm sure I'm going to get even more in Poland next week. And so I have assignments for people here too, so be ready on this. Anyone who is a speaker has at least 1 or 2 jobs of what they need to do. So we need to complete and understand the pathways for lutein and zeaxanthin uptake from the diet and into the eye. Clearly I'm involved in that. John Landrum, Wolfgang Schalk, Liz Johnson, see if I can remember everyone who is on here. I think it's very important. We've learned a lot about mesozeaxanthin, and I'm going to take it as a personal challenge. 2 years from now, I hope to be able to tell you what the enzyme is and how it all works. So we need to elucidate the mechanisms for enzymatic or non-enzymatic production of mesozeaxanthin and other metabolites and define their roles. And I think John Nolan, obviously, because he's working on mesozeaxanthin, has to do that. And is it David Thurman? Okay, I'll get the name right. We, of course, need to define the mechanisms of carotenoid function in the retina and brain. Liz Johnson, Randy Hammond, and who's our B? Richard Bone. Okay, yes, of course, on that. Sorry about that. And we have to continue and improve and utilize the non-invasive methods of carotenoid assessment. And there's many people. I have just a selection of 4 people who should be doing that. And finally, this is a big one, and a lot of us are involved in this. We need to work on clinical studies, the epidemiology, the genetics, get KARMA published, get AREDS2 published and out there. And that's, there's many of us involved. I'm involved as an AREDS2 investigator. So hopefully we will have, in 2 years, a lot more to say and a lot more information. And I, of course, want to thank, again, everyone who's been in my laboratory and my collaborators, my major collaborators, and the people who have funded me. So thank you.
There you go.
Okay. So
Hold on, your mic is after.
Okay. All right, so it's time for grades. Fortunately, I'm not going to do that. At least I'm not going to rank every people. I would say that we as a group still get an incomplete. We're not quite there. I'm not going to give you a fail. I'm not going to give everyone a pass. But we did. It's been a wonderful conference, but we still have much more to do. This was the homework from before, and it hasn't changed dramatically. That's what keeps us in business. That's what keeps us working here. We still, and I've added some people have been added in, some people have been removed, and we've added a few more points of what we need to do. But still, we need to look at the pathways of lutein and zeaxanthin. But this time I've actually expanded a little bit, make it a little more. We have to go from, not only just from the diet to the eye, but actually from the garden to the eye, as we've heard from Tim O'Hare. Did I get that right? Yeah. From Tim about improving our crops. And that's one of the things that will come in next year's meeting, a year from now. We will have many more plant people there. They need to understand what we need as in the carotenoid field, what we need for clinical work and how to improve plants and how to improve production and how to learn from each other. I would say that in this meeting there hasn't been a lot about dietary uptake, absorption from the gut. That's something that I'll try to incorporate a little bit more into the meeting in Park City. That kind of is the missing thing. We just kind of say, well, you eat it and it gets into the bloodstream. Well, we know things are very different. Fred was mentioning today just, the ratios that we eat in our diet are not the same that we see in the serum. Beta-cryptozanthin is really bioavailable. I mean, it's very high in the serum. We don't eat that much of it, but it's consistently there. Astaxanthin, really, some people eat a lot, but you rarely ever see it in anyone's serum. So there's a lot of differences that we need to understand. There's a lot of interactions with retinoids, and so that's something we need to learn a lot more about. Obviously, there's a lot of work on mesozeaxanthin and their metabolites and to define their roles. We've just spent a long time discussing that. I clearly have not solved the enzyme for figuring out mesozeaxanthin, so I'm asking for a 2-year extension, a 1 to 2 year extension from the group here. But Fred Katrick and I are very actively working on this, and we have some ideas, and hopefully we'll know more. We need to define what are the substrates, what are the enzymes, what is the significance of this pathway. Clearly, and I kind of expanded here, the third point is define the mechanisms of carotenoid function in the retina and the brain from infancy to old age. I think there's been a lot of interesting work that's been presented here that carotenoids are not just for AMD anymore. They're for the entire lifespan. And for many other tissues. They're not just for the eye anymore, too. And that's a very exciting area. And at the International Carotenoid Society, we'll try to even expand and their roles in the prostate, in cardiovascular disease, everything that's out there. It's a very fascinating field. In terms of measurement devices, I'm very impressed at the number of new devices that are out there, and people are still working on both the psychophysical and the optical type methods of measuring macular pigment. There's a role for almost everything, even with the controversies of Raman spectroscopy. You can see it's even coming back, and people are trying to improve what we did just 10 years ago in developing Raman spectroscopy. There was a very good poster on that. And so all methods provide us interesting information that's very complementary and can be used. And clearly, it's becoming very important to understand the role of distributions, and that's something that we need to understand why these quotes atypical distributions are occurring, what their significance is, and what the possible ways of altering them for better or worse are. And we need to understand how supplements influence that. We've made a lot of progress on the clinical studies. Some very important clinical studies have come out, many of them just in the last few months. And so they are not perfect, but AREDS2 is providing guidance of what we will be doing in clinical practice. The KARMA study has come out, and there's clearly much more to do in epidemiology and genetics, and more clinical studies, large and small, will be coming out, and we need to learn from them. But I don't think there will be anything as big as AREDS2. There will not be an AREDS2. We can't afford, unfortunately, to do the really big studies. But we've provided a lot of good information to guide people. We heard this morning, I added this morning, interesting work from Toss Barenschot on xanthophylls and inflammation. That's going to be, as he said, it has to be replicated, it has to be understood, and we need to go forward on that and see what's going on. Essentially, looking at the effects that are occurring systemically from consuming carotenoids. And that can even be emphasized when I was talking about the problems of mouse models of carotenoid function. We know, as I was able to show, that the carotenoids in a normal mouse don't get into the eye, but people are publishing many, many effects of administering carotenoids into the mouse. So clearly other things are happening in other parts of the body, metabolites are happening, and we need to learn from that. And then there's, which kind of goes with the clinical studies, we need to learn, we've heard some very interesting work on AMD and macular pigment and genetics, the role of genetics in this. Now that we understand, as we're understanding more and more of the biochemistry, understanding the proteins, we could look to see their relationship to macular pigment distributions, macular pigment optical density, and AMD, and try to look for their derangements and see what we can learn from this. Plus, we may even learn more. I think Julie's not here anymore. Yeah. But in Julie Maris, we're in the era of GWAS, gene-wide association studies, where you don't have any, your only hypothesis is that a gene may be associated with something, associated with macular pigment. And she's been finding many unexpected genes, and we need to understand, is this kind of random associations, or is this real? Are there other pathways that we haven't understood that are associated with AMD, associated with carotenoids? So we clearly have lots of work to do, lots of work to keep us busy, and lots of reasons to come back together at meetings 1 year from now in Park City, 2 years from now here. So thank you very much.
So I suppose that concludes, and I just once again like to thank the Howard Foundation, the many sponsors, all of you, and I wish you all a very safe trip back. And as Paul said, we wish you the best with your scientific studies. We look forward to seeing the results of those. Thank you very much.